mass, but pretty much only the heart and some of the excretory system sits in there. We'll look at that in a little more detail. The exterior dorsal side often has a shell. And in fact, in the sort of basic setup, there is a shell that's on the outside. Okay, that's it. There's the basic body plan. Visceral mass, foot, that little space in the back where the gill is at the back is called the mantle cavity. The mantle is the exterior part of the shell that makes, or exterior part of the body that makes the shell. Have a little head up here. That's it. From this little simple plan, all of the kinds of mollusks that we can see can be easily modified from this simple plan. Here's the thing that's really mind-blowing. This mollusk doesn't exist now. There is no mollusk that actually looks like this. In fact, we call this thing the hypothetical ancestral mollusk. It is a body plan that could have existed from which all of the modern mollusks arose. But currently, there is no mollusk alive that actually has this exact setup. Everything that we see in modern day mollusks is a slight modification of this. From this little basic plan, mollusks have been able to invade every single part of the planet except the air. There are no flying mollusks. It'd be kind of cool if there were, but there aren't. They live freshwater, marine systems, land, but they don't fly. So interiorly, all of the parts are a little bit more complex than just a visceral mass and foot. There's a linear gut. There is a heart, coelomic space around it, musculature that attaches to the shell down to the foot, some associated mouth parts, and we're going to talk about those in more detail. So the coelomic space develops as a schizocele. What does that mean? As opposed to an enterocele.
Spaces appear independently of the mesoderm. Is that right? Wait.
They
Appear independently of the mesoderm.
They appear independently within the mesoderm. They are not pinches
Oh,
off
and
it's not an enterocele, it's not a pinch off of the developing archegantron. Okay, that's good. In the visceral mass, that's where all the organs are found. The foot is muscular and is often covered with cilia, and it is the main locomotor organ. So they might either, in the most basic setup, it's a creeping sole. There's little muscular undulations that help you sort of creep along the surface of whatever it is you're on. It secretes a mucus layer to help you do that. On the dorsal side is the mantle. This is the mantle just beneath the shell. The mantle is what secretes the shell, so it's like an epidermal layer that has the ability to secrete calcium carbonate. And that calcium carbonate gets laid into a matrix on a cellular level that forms the shell. That shell protects the animal, but in the most basic setup, the animal can't actually pull into the shell. They can maybe clamp down on a rock, and that's what we think the ancestral setup was. An organism that crawled along hard substrates and was able to use the muscular foot to clamp down onto the rock and hold themselves onto that hard substrate, protecting themselves from some other predator. One of the most cool developments in this group is the formation of a set of mouth parts that help them feed in a very special way. So that front mouth part is called a radula. The radula is like a file-like tongue. It is able to scrape surfaces with the file-like tongue and pull off things that usually like biofilm layers of bacteria or algae, and scrapes it off and moves it into the mouth. Is able to move it through the esophagus and into a gut where digestion takes place, and ultimately can use the length of the linear gut to process along the way. So the way that they do this is really amazing. So this is a scanning electron micrograph of a radula over on the right-hand side. It literally looks like a file, and a recent paper came out about 1.5 years ago that suggests that the radula is the hardest biological made material. Harder than steel. It's a scleroterized proteinaceous setup, so it's like embedded iron inside of a proteinaceous matrix that makes it super hard. So this thing scrapes along the surface and rips up particles that are edible and slides back and forth over this sort of like this sort of rigid, it's called the odonophore. So radula is like a belt that slides over the odonophore and scrapes up things and moves it into the mouth. But it doesn't have a musculature around the mouth. It's not actually able to swallow stuff. So how does it get food into the mouth? This is not something that's displayed here. I'm just going to tell it to you because it's so bizarre. There is a gland that secretes a mucus string. And that mucus string runs all the way back through the esophagus and into the back of the stomach. At the back of the stomach, there is a hardened spike called the crystalline style that's sitting in a cilia-lined pocket. The cilia beat and turn the crystalline style. The crystalline style pulls the mucus string from the back. So as the thing licks with the radula, particles of food get embedded in the mucus string and get pulled to the back of the stomach along the mucus string by the spinning crystalline style. Okay, here's where it gets really weird. The crystalline style pulls the mucus string into a hardened ciliated surface. It's called the sorting field. And as the style scrapes along the sorting field, the style is actually made of digestive juices that digest the mucus string, release the particles of food into the sorting field. The sorting field, all the cilia, picks out the food particles that are edible, move them into the stomach or move them into the digestive ceca. The things that are not edible shifts them over into another set of mucus that then get ejected as a pellet. Incredibly complicated setup for swallowing food and digesting food. All that specialization, though, allows it to continuously feed. It is always pulling mucus back, always scraping that mucus or scraping the crystalline style across the sorting field, always being able to digest more and more food. And if you think about some of the more simple organisms we've looked at, like cnidarians and cloudy helminths, they cannot continuously feed. Once they ingest stuff, they have to stop, digest it, and then remove the non-digestible material before they can feed again. So I like to think of these guys as sort of like little tanks that sort of work their way along the surface of the substrate, licking up food along the way, and will just form like a little, shredded patch where they've moved along and just fed continuously. So the radula is really cool. We'll have one for you to look at in lab today, because the radula is actually very closely associated with what kinds of things can be fed upon. So this is a radula that has a central groove, but there are some with multiple grooves. There are some that are like a single layer of teeth. There's one mollusk that feeds on plants by cutting open the individual cells and sucking out the insides, and so it does it just by like a slice that's done by a saw-like radula. The radulas can get modified into all kinds of weird things. There's a whole group of snails that have a radula that's been modified into a hypodermic needle that injects incredibly complicated venoms into hosts or into their prey. Cone shells. You thought that box jellies were badass. Cone shells kill more people than box jellies. Yes. More people are killed by snails in a given year than by sharks. Those snails have an incredibly strong neurotoxin. And so what happens is not that they get hit by the snail and they die. They get hit by the snail, and the snail's neurotoxins basically freeze up your spinal cord. So when you see fish get hit by this, they have like this 10-second shot where their whole spinal cord goes, and then they sort of flop back and forth, and then they turn to the sides. So anyway, when humans get hit with this, they basically freeze up for 15 or 20 seconds, fall into the water, and then can't get out of the water and drown. So they die from drowning, but they get hit. It's a snail that makes them do it. The guy who studies these things, he's at the University of Washington, Seattle. He studies cone shells, C-O-N-E shells, and honest to goodness, his name is Al Cohn, K-H-O-N. He's the guy, he's the top guy in studying cone shells. His name is actually Cohn. All right, so feeding modifications. We also see circulatory modifications in this group of animals. They have an open circulatory system, which means that the organs are within an empty space in the visceral mass that's not a coelom. It's just this, it's what we call a hemocoel. It's an open space that the fluid bathes each of those individual organs. So it has a complicated two-chambered heart that pumps fluid into that open space and picks it up from the other side of that open space. So those hemolymph have cells that are able to carry oxygen. So there are some that have hemoglobin and hemoglobin-like compounds. The more advanced mollusks, the cephalopods, which are squid and octopus, those actually have a closed system that's much more like yours and mine, where there is a heart that pumps through fluids that have, through arteries and veins that drop down to capillaries and supply individual cells with nutrition and gas. We'll see that again a little more in a little bit. Excretory systems are one of the most exciting big changes in this group. The coelom is reduced and surrounds around the heart, but that coelomic space is used as a location of sequestering nitrogenous wastes. So nitrogenous wastes get picked up in the hemolymph and moved back through the heart. And the heart sits inside of this cavity. So here's your heart sitting inside this cavity. Sorry, here's your heart sitting inside the cavity. And the pressure of the heart pushes fluids out through these little ostia on the side of the heart and into the coelomic space. That coelomic space is then drained of those nitrogenous wastes through a cilia-lined duct that's called the nephridiophore right here. So we have a nephridium, which is a structure, this is your nephridium, which is a structure that through the pressure in the hemolymph pulls the nitrogenous wastes across a filtrating membrane and into a sequestered space, which then drains out through a pore to the exterior environment. And the cilia-lined pore is set up in such a way that it can reabsorb important nutrients, it can reabsorb important salts, and can limit the loss of things that the organism needs. So much more complicated and advanced than what we see in things like the platyhelminths that just have a flame cell that drains those nitrogenous wastes directly out through a pore. It has this complicated set of membranes that help sequester and concentrate those nitrogenous wastes for removal from the organism. So that structure is called the nephridium, and the way that it moves out is the nephridiophore. Respiratory systems are a lot more advanced in this group as well. When we saw things like the platyhelminths, how did they respire? What did they do for gas exchange? They have any specialized structures to do it? So how'd they do it? Diffusion directly across the body wall. That means no part of the animal can get big enough that it can't be, like as an animal gets bigger and bigger, it has to have certain ways it's made to continually be able to supply those interior cells with gas. So how does a flatworm do it? If they don't have any structures to make it so that they can get that gas, how do they do it? There's a hint in the name of the animal. It stays flat. So no cell, even in the middle of it, is very far away because it's a flat structure. These animals can get quite large. Mollusks can get really big. And that means they need a specialized structure to be able to do that gas exchange. They do it by forming a they do it by increasing the surface area of one particular structure that allows for lots and lots of gas exchange to take place. And by running that hemolymph through it, the gases can diffuse across the surfaces of those structures into the interior fluid, and then the fluid gets carried around to the rest of the organism. So the circulatory system is coupled with the respiratory system. So in this case, what they have are gills. These are gills. And gills are, in mollusks, we call them ctenidia. Remember, tenes are combs. So we saw that in ctenophores. It's the same word, ctenidia. They're comb-like structures that are used for gas exchange. So what that means is that the surface of the ctenidia are incredibly thin membranes, easy for gas to diffuse across, directly into the fluids that's inside. But those fluids can get moved to the other parts of the body. What that also means, and when you think about what happens with diffusion, if you have a surface like that that's diffusing gases away from the fluids exteriorly, unless you replace the fluids exteriorly with new fluids that also have high levels of oxygen, diffusion is going to come to an equilibrium and not flow through very quickly. So while moving those gases away from inside of the body of the animal, it also has to replace the fluids outside with newly oxygenated fluids. And it does it by having cilia all over the surface of those ctenidia. The cilia beat and form a current. And so you actually have a current that comes into the mantle cavity through an in-current siphon, goes across the gills, exchanges oxygen, and then goes out an ex-current siphon. We see it in the hypothetical ancestral mollusks by the gills having cilia on them and making a current. Flow comes in across the gills and then out the mantle cavity. The way that this works to be able to maximize the diffusion of gas is that the water from the exterior goes in one direction and the blood from the interior of the gills goes the other direction, and you get a cross-current flow, where that makes it so that you can get the highest possible amount of oxygenation in the blood. So, for the most part, their reproduction is separate sexes or dioecious. There's males and females, although there are some groups that have hermaphrodites. That fertilization is generally exterior, so you put sperm and egg into the water column, and they fertilize in the water column, although there are many that do have internal fertilization. For most aquatic mollusks, they fertilize externally. If you're in the water column, it's easy just to put your sperm and egg up in the water column. But there are many that do have internal fertilization. Some of them in really interesting, weird ways. For example, cephalopods make a spermatophore. They actually make a little packet of sperm, and the male octopus or squid uses a specialized arm to grab hold of their own spermatophore and place it inside the mantle cavity of the female. So the larval form starts as this what's called trochophore larvae. It looks like a little spinning top, tufts on the top, and this sort of band that feeds. And in mollusks, they develop, at least the pelagic ones that live in the water column, they develop this big feeding appendage on the way that's called a velum. So they get the name the veliger larvae. And so they've got this big feeding appendage that's covered with cilia, and they swim around and collect the food that's in the water column and feed upon it. So, okay. Notice that each one of these things we say, this is the basic sort of setup, but there's always modifications and differences. And that's because these things have been able to become incredibly diverse in their forms and structures just by modifying the sort of basic setup of each individual one. So how can we get from this to each of the forms of mollusks that exist out there? So I'm going to start with the first one and try and use the same colors so that you see what the setup is here. Black and, okay, so this is our, we call this ham. What did ham stand for, if you guys know? Hypothetical ancestral mollusk. All right, so let's see if we can get from ham to the chitons. So I'm just going to push it right over here, okay? So for chitons, we still have our visceral mass, and we still have our head, and we still have our foot. And we still have our foot. Okay, so we got the parts. But in order to make it a chiton, chitons have a series of 8 shell plates on their backs. So we're just going to put little 8 plates. 1, 2, 3, 4, 5, 6, 7, 8. And rather than have the mantle be posterior, the mantle is just along the side of the foot. So I'm going to do that in red. Set of gills running along the side of the foot. That's a pretty easy modification to make to the hypothetical ancestral plan to come up with the body form for that group. Piece of cake shift, okay? All right, what about, there's a group called aplacophorans, not up there. What do you do for aplacophorans? You just get rid of the shell. It's the only group that actually has the mantle posteriorly. So, all right, what's the next one we want to talk about? Let's talk about gastropods. All right, so gastropods are kind of cool. They look just like ham. Okay? They got a visceral mass. They've got a head. They've got a foot. But for mollusks, or for gastropods, they actually have a very simple modification. Rather than having the mantle cavity be posterior at the back of the animal, the mantle cavity ends up anterior over the head of the animal. There's your gastropods, snails and slugs. All right, what about the next group? Let's do bivalves, because bivalves are kind of easy as well. So bivalves, okay, still the same setup. They got a visceral mass. The mantle cavity actually runs the entire central part of the organism on either side of the foot. But the foot gets modified to be a blade-like structure, and the head gets reduced. So it basically doesn't have a head. The mantle cavity ends up running on either side of the foot, and so you end up with your gills or your tibia running on either side of the foot. And the shell ends up forming 2 lobes on top of the foot. There's your bivalve. Okay, now the last one seems a little bit tough. How do you get a squid or an octopus out of this basic plan? For that, all you really need to do, and even though it looks like it's pretty crazy, all you have to do is take the entire animal and push the visceral mass up super high. The mantle cavity still exists in the same spot that it did. Oh, sorry, I put it in the wrong color. This needs to be red. The mantle cavity still exists in the same spot. The head still exists in the same spot, just right here. And the foot becomes a series of modified tentacles hanging down. Whenever I see movies where they make the animal, you always see this, right? The squid, it looks like he's got this big giant head and then a couple of tentacles, right? Or the octopus, that big thing that looks like the head that has two eyes, that's actually the body. If you just turn this thing to the side, drop it down like this, you turn this thing to the side, you've got your tentacles, your mantle cavity, and your body, and your head. That's all there is for the head. Just that little bit. All of this stuff is actually the body. So I have a hard time watching like any movie that represents octopus and squid, where they treat that big visceral mass as his head. Just freaks me out. Like I can't watch Finding Nemo because every time it's like anatomically incorrect cephalopods. Drive you crazy. All right. So, even though the incredible diversity of cephalopods, or the incredible diversity of mollusks, seems like they're really divergent in their body plans, they're all just modifications of the hypothetical ancestral mollusk. They're all just slight shifts in the way that thing is set up to form individual body plans. So, the main representative groups of mollusks, we're only going to talk about 5 big guys, but there's a bunch of others. There are aplacophorans. Aplacophorans are really easy. So aplacophorans are really easy. They still have a head. They still have a foot. They still have a visceral mass. They just don't have a shell. Right? Same sort of setup. The other group that we're not going to talk much about are the Scaphopoda. The Scaphopoda still have a head, and their visceral mass ends up forming in a little tusk-looking like thing. The mantle cavity runs up along the side like this, and they have a foot that looks almost kind of like a, right? They look like that, the tusk shell, Scaphopoda. Monoplacophorans. Mono means one, placophoran means shell, so we got one shell. Look like a little shield. They almost look like the hypothetical ancestral mollusk, except their gills are set up along the side of the body the way that it is in the Polyplacophora. That's the chitons, the Polyplacophora. Gastropods are snails and their relatives. Bivalves are clams and their relatives. Cephalopods are squid and octopus and their relatives.
Wait, what'd you say chitons are?
Chitons are polyplacophorans. That's the class.
Example.
What's an example of them? Chitons.
Thank you.
Well, I'll show you a picture.
Okay.
All right. How about that? Okay. So, monoplacophorans are actually kind of cool because we know them from fossils, and up until the mid 50s, we thought they were a extinct lineage. So in the 1950s, we finally collected some specimens from deep sea, and they look kind of like these limpet flat shells, but if you look close, you'll see there's this series of gills running down the side of the foot, and so even though they're round, and even though they have these set of gills on the side of the foot, they actually look a lot more like polyplacophorans now that we have live specimens. When we've looked at molecular setups, where do you see the monoplacophorans here? This is right through here. Can you tell that tree from here? Okay, so green are the chitons, and that pink is monoplacophora. The monoplacophoran specimen here actually sits within the polyplacophorans. So even though we're still using monoplacophora as a class, it appears to be monoplacophorans are just a specialized kind of chitin, just a specialized kind of polyplacophoran whose shells have all fused together. I know. so these guys are all deep sea. They have a body setup that's more like a Polyplacophora now that we actually see them. And when we look at what their interior structures are, they're essentially Polyplacophoras. Okay, so here's your Polyplacophoran. Have you ever seen something that looks like this living on a rock? No? Okay. Polyplacophorans have 8 interlocking shell plates along their exterior. They're surrounded by this little girdle that allows them to clamp down onto rock structures. So they actually can form like a little suction cup, right? They like clamp down and then lift up their foot below, and it's the same thing as a suction cup. So they often have a single spot on a single rock that's their home spot. And they sit there until they go out and feed, and they go on this little foraging trip that they go and feed, and they come back to the exact same spot on the same rock. For the most part, they're dioecious, so they have separate sexes. They have a radula. They're very much like what we think that hypothetical ancestral mollusk might have looked like, except a single plate and a mantle cavity that runs down either side of the foot. And they also have a set of serial repeating gills along the side. And those gills are linked to musculature that's attached to each plate. So it almost looks like a sort of specialized segmentation beginning. Remember segmentation, serial repetition of the body parts, right? Serial repetition of individual gills that are associated with serial repetition of musculature attached to each of the serially repeating shell plates. Polyplacophora. All right, when you guys think,
Why do some of them have spikes?
why do they have spikes on them? A lot of them have spikes on the girdle. So the girdle is not actually a shell. It's like a proteinaceous sort of like almost like keratin, almost like fingernail kind of stuff. And those spikes do really good at having predators not be able to take a bite out of the side of your girdle. Yeah. And much of these guys, they spend their lives attached to rocks, sort of just hoping nobody eats them. But there's lots of variation in the girdle and thickness in the girdle. And you're going to look at some of them in lab today.
So if you put it on your hand, would it suction to your hand?
It might try to, yeah. Might be. I have a friend who's he's at UC Fullerton, who's one of the only guys who's really working on these a lot, named David Henderson. And so he's a Facebook friend, and literally every day he posts pictures of penguins on his Facebook page. They're kind of cool, and they're very understudied, and he's found maybe 15, 20 new species in just the last few years for molecular means. In a place like Southern California that's incredibly well sampled, he keeps finding new stuff that people haven't recognized as being new species. All right, so when you guys think of mollusks, no doubt you think of things like slugs and snails. Snails are the biggest group of mollusks, about 35,000 named species, but probably there are a lot more. They live in freshwater marine systems. They live on land. You guys have seen land snails, right? In land snails, it's actually really cool. Their mantle is folded over and forms an air-breathing lung. So have you ever seen like a slug sort of going along? Look at the side of his head. He's got this little hole on the side of his head. It's called a pneumatostome. That hole leads into an interior air-breathing cavity, basically a lung. But the question is, why does he have a hole behind his head as the location where entrance to the lung is? The reason is that snails have one trait that keeps them all in common. If you notice in our little body plan here, the posterior mantle cavity ends up over the head. That trait for snails is what unifies the entire group of gastropods. It's called torsion. In torsion, one slide ahead. Okay, in torsion, the whole entire visceral mass turns 180 degrees during development. So the larvae is born like a normal snail, And as it develops like this normally. And then at a certain point during development, the entire visceral hump, the entire visceral mass turns 180 degrees during development. Which means you now have your mantle cavity on top of your head. So all of the things that are included in the mantle cavity, like what? Your anus. So your anus comes up like this. What color is it? Your anus comes up like this, does a little loop-de-loop because it's actually turned around during development, and comes back to here. Okay? So what does that mean for pooping? What's that? So here's your head. Here's your pooper. It poops on your head. So that's kind of a tough thing if you're a snail, right? Especially since if the flow of this works exactly the same as it has, right? You'd come up and poop right on top of your head. Okay, so that's a problem. But clearly it didn't hurt snails because they're the most diverse of the group, right? All right. Also, if your nerve endings go like this, down your legs, and in this case, the nerve endings go down your legs here and then up into the visceral mass, when this whole thing gets turned around, you end up having this have to do a figure eight. So the entire structure inside the visceral mass is all rearranged because it does this 180-degree turn during development. It does this little flip during development. Have you ever seen a snail or a slug? The reason why they've got that hole above their head is because their breathing apparatus is above their head. That also means that they poop out of that hole. They leave nitrogenous wastes out of that hole. So they've actually gone through torsion and then sort of like half undone so that it's all on the side of them, not directly over their head. There's also groups of snails that go through full detorsion. They grow, they tort, they put their mantle cavity over their head, and then as they continue to develop, they detort back again. And in some of those, you get a double figure eight. The nerve endings goes up, rotates back, and then flips again back because the detorsion doesn't just go back the same way. It goes all the way around. Totally weird-looking things in these groups. Torsion is what makes a snail a snail. This is what makes a gastropod a gastropod. The fact that the mantle cavity has shifted from being posterior to being anterior. But they all still have the same sort of things that you see in other mollusks as basic parts of the mollusk body plan. They've got trophophore larvae. Many of them are monoecious, especially freshwater organisms or terrestrial. There's some really cool mating behaviors that take place with some of the terrestrial slugs. They'll do these things where like they climb up a tree and then secrete a mucus string, and the two slugs hang down the mucus string, like hanging off of the tree, and they mate while hanging off of the tree in this little mucus string. Those are kind of cool. There's lots that have modified radulas for specific things, including things like the cone snails that are used as a hypodermic needle. The coiling shell of a snail is not actually related to torsion. The coiling of the shell and torsion are separate phenomena. So they're not like, coiled shells were not a response to torsion, and torsion wasn't a response to coiled shells. We're actually not sure why torsion took place. We have some good stories about what could have made it be more advantageous, but we don't really know evolutionarily what was the thing that made torsion be so advantageous to those early mollusks. But it must have been incredibly advantageous because modern day gastropods are by far the most diverse of all of the groups of mollusks. So this is the class I work on, bivalves. Bivalves take your ancestral mollusk, split that shell in half with a seam in the middle, fold it around the whole animal, lose your head, move your mantle cavity to either side of your foot. Bivalve means two shells, so all of these guys have two shells with a hinge or seam down the back. They include clams and oysters and mussels and things like that. Over 10,000 species described, but those numbers are probably an undercount. Those of you who have seen any of the work of my students, we are working on a Brachydontis exustus, a single named species, but we're up to about 8 species within the cryptid complex, and that's probably not all that uncommon. There's probably lots of named species that represent a lot more species than that. They come from marine systems, freshwater systems. We use them as food. There are some that are incredibly problematic as pests. Anybody recognize this guy? That's the zebra mussel. Zebra mussels were introduced into the Great Lakes in 1989 and became a tremendous problem for things like water intake systems, pretty much any hard substrate, plus our local freshwater bivalves, the unionic clams. Those zebra mussels land on them and basically outcompete them. So in a place like Lake Erie, they went from not existing at all to being at levels of around 10,000 organisms per square meter, spreading out across almost all of the lake. So they actually completely changed the ecology of the lake. Instead of having food webs taking place up in the water column, the zebra mussels just eat everything. And the water's a lot clearer, but there's a lot less diversity.
I was gonna see, is there, do they, I don't know, well, we go to this place in Tennessee every year. Now they have it because like people in Cincinnati brought it from Lake
They're
Erie.
spreading everywhere, yeah.
They like the water's cleaner now.
Yeah.
Is that because of
An individual, so each one's about the size of your fingernail.
razor sharp?
Well, they hurt if you step on them, that's right. Each individual one of those dudes can filter about a half a liter of volume through their bodies in a day. So if you have 10,000 of them per square meter, a single square meter can filter 5,000 liters, or it's about 1,200 gallons. So if you have a water column that's, let's say, 10 feet deep, that is less water, or that's more water than in the direct column over that meter. So in a place like Lake Erie, every molecule of water in Lake Erie makes it through the mantle cavity and filtered by a zebra mussel, usually in less than a 3-day period. So think about it. Any phytoplankton that grows, if they can't make it to maturity in 3 days, they can't reproduce enough to maintain their populations, and they get eaten out by zebra mussels. So zebra mussels are nothing, though. There's another one coming on its way that's coming up from South America that will eventually invade every freshwater body on the planet. It has a much broader ability to grow under low calcium that zebra mussels do. That one is called Limnoperna fortuni. And in places where zebra mussels were already established, called the golden mussel, the places where zebra mussels are already established, when Limnoperna gets introduced, Limnoperna can reproduce at high levels with even less food than zebra mussels. So when they get introduced, they outcompete zebra mussels and work down the biomass. So when zebra mussels show up, they change the ecology, but there's still a lot of biomass in zebra mussels. When Limnoperna show up, you get even less biomass, less biological activity. Basically, the only thing that's there are Limnoperna. And they're coming. Not yet in North America, but they made it to South America about 15 years ago. They're pretty much all the way up through the Amazon and near Panama now.
Why do you sound so excited for something that sounds so bad?
I'm not excited. It is bad, but it's a sign of how things are happening in the planet. We're moving organisms around all the time, and things that have been able to be, have their populations kept in check where places where they were once, now get into new places and can't be. They lose their natural predators. Part of the reason why I'm excited is because as an undergraduate, I had to do a senior project, right, a big project, and I did it on zebra mussels. So this was in 1994. They'd been introduced in 1989, so they've been around for less than 5 years. And I made a map of where the expected places that they would show up by at that point it sounded so far off in the distance. The year 2000, it's 15 years away. It's forever away. But, or 6 years away. But it was like forever away, right? And my model worked out pretty well. They pretty much moved as far as I thought they would back then. So together, right, is the valves have this one muscle that sort of clamps it down. So when the muscle relaxes, there's a hinge that has a ligament that opens them up. And so when they're alive, they can clamp down shut. When they're dead, they open up. So if you've ever wondered if you've ever gotten like mussels from the store before you cook them, you're supposed to take the ones that are already open, you tap on them, and if they shut, they're good to go. And if they don't shut, you don't want to eat them, because the ones that don't shut are already dead. They start to break down incredibly fast. They go, I don't eat these things. I think they taste horrible. So, you look inside, you've got your foot, which acts like this sort of blade-like structure that can push down into sediments, flare at the end, and then pull the rest of the animal down below the substrate. There's gills, but those gills are wrapped in a mantle with an incurrent and excurrent siphon. So a lot of these guys actually dig their way into the sediment and just stick their siphons up over the top of the surface of the sediments. Water flows in, goes across the gills, and flows back out. But they're protected by being under the sediments. The two big adductor muscles here, 1 and 2, hold the thing in. Their heart is sitting within the coelomic space. Here's their anus at the excurrent siphon. Notice the head has been totally reduced. It would be down at this end if it existed, but it's totally gone. So, most of them are dioecious, male and female separate. A lot of them have what are called sequential hermaphrodites. So like oysters, when they're born, they spend the first 3 or 4 years as males, and then after their 3rd or 4th season, they shift to females. Their gonads become ovaries, and they start making eggs, and then they finish their lives as females. The gonads are in the visceral mass, and they empty into the mantle so that they'll spew water into the water column, gametes. So sperm and egg just go up in the water column. Fertilization takes place in the water column. They swim around as veliger larvae for a little bit, and then settle down to the substrate for another round. In freshwater systems, has anybody ever dug around any of the local rivers or lakes around here and found a clam-looking thing? No? You guys have one of the most diverse groups of biological fauna in this part of the world. There were over 400 species of freshwater clam right in the Midwest. Illinois had 100 of them 100 years ago, 100 different species of freshwater clam. In the last survey in 2012, they found 46. So we've lost about half of the species in Illinois already. We've lost about half of the species nationwide. We're down to around 190 out of the 400 species or so that they were. What's really cool about our local freshwater clams is that they have a parasitic larval stage that uses a host fish. So the way that they work is they got these little baby clams that have little baby clam shells, but these hooks and spikes on the end that clamp onto the gills of a fish and suck the blood of the fish. So little parasitic larval stages. And what's really cool is that a lot of these clams have body parts that look like fish. They've got little mantle flaps that look like swimming fish, or egg cases that look like fish. And when the fish comes down to feed on it, they get a face full of these little larvae that are called Glochidia, breathe it in their mouths, when things clamp on their gills, and then start to suck their blood and grow up to be a certain size on the gill and then drop off onto the bottom. So we've lost a lot of the freshwater species because if you do something like dam a river, the host fish can't swim up and down the river anymore. And now the clam has no hosts. So some of the clams that are still around exist in incredibly small populations in incredibly restricted areas. So just a couple months ago, Illinois shifted an entire species in one river 6 miles. There were 5,000 individuals at a spot where the Department of Transportation decided to build a bridge. We couldn't convince them to move it a half mile up or downstream. This was the place they had to build the bridge, and it's the only place this species exists. It exists in a 200-meter section of river, and that's it. This little riffly area, 200 meters along the river, and that is all for the entire species. And the Illinois Natural History Survey went out and collected every single clam and moved it a few miles downriver to a new spot. We have no idea if it's going to be successful or not. But that's how tenuous it is for a lot of these species. They're stuck in just little tiny patches, and that's it. The last class are the cephalopoda, everyone's favorite. Cephalopods are awesome because they are by far the most intelligent of invertebrates and are really complex, especially because their body setup is one in which the nervous system is actually a little bit decentralized. So even though they're really smart, this is not their head, by the way. Remember, their head is not this whole thing. Their head is just this spot right here. Their head is just this spot right here, or this spot right here. Well, first of all, they've got fantastic eyesight. Some of the best eyes on the planet. Their eye does not have the same problem that your eye has. You guys know that your eye is set up in a way where the nerve endings come through the back of the eye and then spread out the front. You've got this blind spot right in the middle of your eye. Like, you can actually goof around with your fingers and sort of get them into the blind spot where you're actually only seeing each of the fingers from the eye. Yes, you have a blind spot right in the middle of your eye. And your brain just sort of fills that stuff in. It doesn't really know what's there. It just sort of fills it in and makes something up so that you don't recognize that there's this big hole right in the middle of your field of view. Mollusks don't have that. They've got it from the back. So their retina covers over the whole back and they can have a full field of view. How do you guys deal with changing your focus? What does your eye do to change your focus? Blink? No, like you can see really far away, but then you can moments later see really up close. How does your body do? All right, who wants to be an optometrist? Well, your pupils dilate. That lets in and out light.
Your lens shifts and it either like expands.
You've got muscles around your lens that shift the lens, right? So many of you have setups where the lens doesn't actually focus on the back of your retina. Who wears glasses? So think about how shitty your eyes are if half the people in the room can't actually see without mechanical help, right? Literally half the people in the room, your eyes are defective, and I'm not saying this to be nasty or mean. They don't work right. They don't focus the image on the back of your retina so that you can actually see. You have to put something in front of it so that your lenses that are missing the retina can be compensated for. The way that mollusks do it is they actually have musculature on the entire eye. So they don't move the lens to get the things in focus on the back of the retina. They move the retina. They want to see really close, the musculature pushes that thing right up close to the front of the eye. If they want to see far away, the thing pulls it all the way back. The whole shape of the eye shifts. And they've got fantastic eyesight. There's a reason why you never see mollusks wearing glasses. All right. So, the foot, in this case, is modified into these appendages. But the brain of this thing, notice the brain is right here next to the foot. So the brain is not focused here. The brain actually has these two giant ocular lobes that sit right behind the eyes. And then it has a ring of ganglion at the end of each tentacle. So like half of the brain mass of an octopus is actually at the legs. And that means that each leg has its own little brain and can act independently. If you've ever seen the way mollusks move, right, each leg sort of is working its way separate because they're not coordinated in a unified central brain. They have this ring of subganglion at the base of each leg. So if you cut off a mollusk's leg, if you cut off a octopus leg, it'll still try and collect food and stick it to the mouth that's not there anymore. It'll still crawl around like it's still connected to an organism. Because the central processing section for each individual arm is its own little thing, but unified with the others in a ring, then collected to the ocular lobes. So their brain is not like this big brain mass. It's like these little tiny subbrains all connected together. They're really, really cool. So in most cases, they've lost a shell. Sometimes squid have an interior shell that we call a pen. They have a radula, and that radula is sitting within these chitinous jaws that we call the beak. The jaws actually look like a parrot's beak. And when they open, the radula comes punching out and scrapes through the thing. So like, they're voracious predators because they grab hold of the things with their tentacles or arms, move them in towards where their mouth is, and then their mouth just macerates the thing with this bill or this beak with a radula that's just like rah, 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 and they just shred the things and then engulf. So they're really, really cool. They also have the ability to change the colors of their skin. They have little individual cells that are called chromatophores. There's musculature on those chromatophores. So if you lay a chromatophore flat, it becomes this dark color. If you elongate the chromatophore from the end, it basically disappears. And so they're able to blend in with their surroundings because they can view it with their very good eyes and recreate the patterns on their body surfaces. Some of them actually communicate through those body surfaces. The colors and shapes that shift on their skin are telling other octopus about stuff. So imagine if you could, like, if you were in a good mood and you turned pink, or if you were in a bad mood, you turned like black, right? I mean, it's just be like you could express the way you feel by rainbow colors, or that's what they basically do. They can express the way they feel, and they can talk to each other by changing the colors and shapes of their skin. Okay.
Wait, Dr. Benner, have you ever seen the movie Home?
Have I ever seen who?
The movie Home.
About aliens?
Yeah,
Yeah.
they're exactly.
No.
I mean,
Oh, you mean they explain themselves that way? Yeah. Where do you think they got the idea? I'm telling you,
And
every
they-
single
they
movie
look
they
like
steal from invertebrates.
they have little tentacles on top.
Yeah.
They're little feet.
Yeah.
I forgot what the name.
My kids like that show. So here, check out this little cute guy. Isn't he cute? This is a full-sized adult. This is a full-sized adult. So,
Those are big. You can probably kill somebody.
well, there's one. See how he's got those little flaps up on top? Those are his fins. Those things. So that actually is called the Dumbo squid, because he looks like Dumbo with his little flapping things. So, you guys can read this stuff. I'm out of time. I can just keep talking about them more. Oh, did you guys hear about the squid that you could that picked like 9? He picked 9 World Cup games in a row. They put like the 2 flags, the 2 countries, and the squid would climb out and pick one. 9 in a row. The statistical probability of that squid getting 9 in a row by simply guessing is incredibly low. So Paul, the squid, must know something about soccer. There's even a squid that got caught climbing out. So there was a squid that was being used for, or an octopus